everybody, welcome to class. Thanks for coming. We've got super awesome guest speaker today. We've got Dr. Tom Miller from FSU. He's a biology and ecology professor there, teaching undergrad and graduate school. He's going to tell us a little bit about his research, kind of give us a deep dive into local Tallahassee biology and ecology. So we're going to ask questions during the talk and during the lecture, and then we'll have a little bit of time at the end. So just let me know, let him know, and let's give him a warm welcome, everybody.
So as I explained to the last class, I normally talk to like fourth graders, or I give talks in bars. I mean, that's kind of, yeah. I give talks in waterworks quite a bit. I give talks to like Audubon Society. You guys are someplace in between, so I'm not sure really what to do sometimes. But with those two groups, I'm essentially trying to trick them into thinking about something else. I mean, everybody thinks about carnivorous plants and they go, ooh, that's cool. You know, it eats insects. You know, here's the one most of you probably had, or you may have had or seen, is just a Venus flytrap, right? And so they're just inherently cool. If I go into a fourth grade class, I already have their attention because it's a carnivorous plant. It's cool. So what I used it as is an opportunity to make them think about other stuff, to make them think about things like physiology or evolution or ecosystem ecology. So that's what I'm going to take you guys through. I'm just going to do it in a little bit more blatantly obvious way. And so we can start off thinking about carnivorous plants as a way to think about plant physiology. So why are these a plant? If they eat things, why are they a plant? Why aren't they animals? Anybody know why we consider these to just be plants? Yes? So there is something about the structure that identifies them as within a certain group. There are certainly plants that have different sorts of cell walls. And then there are also some things that are more animal-like that can have things that look like cell walls too. So it's not perfect, but it's a good start. What else? Yes? So they have chlorophyll. So they use photosynthesis to capture energy. You know, everybody needs energy and nutrients. You guys get your energy from eating lots of sugar. They get the energy from photosynthesis, converting sunlight into sugars in some way. And so, but they also need nutrients. You also need nutrients. I don't know where you get your nutrients. You eat fruits and vegetables, right? That's good for you. These guys get their nutrients from capturing insects. And so it's a different sort of process than other plants. But let's consider a few other species. So here we have in the upper right Euglena. And so these are protozoa that swim all around in the water. They're very, very active. You clearly look at them and say, well, that's not a plant, right? It's not a plant at all. And then they have different sorts of cell structure. It's not a plant. And they're swimming around. It can't be a plant. But they're photosynthetic. And so there's lots of organisms that are photosynthetic. There's bacteria that are photosynthetic. There's protozoa that are photosynthetic. But they're not plants. And so being photosynthetic doesn't describe them. This is a Parmelia. It's the same sort of lichen you'll find out on the trees around here. And obviously a lichen is a combination of a fungus and an algae. And so it's not really a plant either. It has some algae in it. So it depends on what group of algae it's in, whether or not we even consider them to be plants. And has anybody seen these before? So these occur around here. I can go up to Maclay and walk around in the woods there and find them certain times of the year. But it's a plant. It's a good plant. I believe these are in the Ericaceae family, so the blueberry family. And they have no chlorophyll at all. They live on the roots of trees, and so they steal nutrients from trees. They steal photosynthate from trees. And so they don't have to be photosynthetic. It means they don't even have to come above ground. The only reason why they come above ground is to flower. And so there's these things that are clearly plants too that we don't really think about the fact that they have no chlorophyll at all. So they don't fit our usual definition of a plant in some way. So it turns out that what a biologist considers to be a plant is something that really, if we take its DNA and we sequence it, it's similar to a bunch of other things we call plants. I mean, it's kind of a stupid definition. It's easier to think about cell walls or chlorophyll because that works most of the time. But in reality, it doesn't work all the time. And so if we just sequence the DNA and then make a big family tree out of it, most of the things that are alive are bacteria. And then we have higher forms of life. Here's us up here in the animals in some way, right up there with the slime molds. And then they're plants. And so it's really based on their genetic relatedness that we're calling them plants in some way. Seems like a weird definition. But they're still green. They have to be photosynthetic. And so why do they capture prey? It says up there too. But why do they have to capture prey? For nutrients. And so it replaces root function. And so they're sacrificing leaves to make these goofy, silly, stupid leaves that might be worse at capturing light, but allow them to capture nutrients as well. And so it replaces root function. Yeah?
So are the root systems just for structural purposes?
And absorbing water. And so they often have pretty minimal root systems. So they don't, some of these actually still do have roots, I think, coming out the bottom. You know, a little bit of roots coming out the bottom. But if you, a lot of potted plants, they quickly have roots coming out the bottom. These don't. Roots grow very slowly. So it really is just to kind of support and to bring in water. Let's see, what else do I have to say? So to capture insects, there are three really, really different ways to capture insects that are kind of fun to think about, again, from the physiology point of view. The first one is to be an active trap, and so that's our Venus flytrap here, which you can see one opening and closing there. And again, I hope all of you have played around with Venus flytraps at some time. Notice it's green. It makes these nice, normal-looking flowers, right? It's just a normal plant, except that it also captures insects. Now, Venus flytrap are native to North and South Carolina. They're endangered there now. They come from the green swamp, and mostly in South Carolina. It's been drained and a lot of housing gone in there, so they aren't as common anymore. This one comes from just west of town, about 35 miles west of town here. And so a long time ago, well, 1985, we actually know exactly when it was, somebody threw seeds out the window of his car, 3 or 4 places out in the forest west of here. And in all the places where we know that he threw out seed, there's now a population of Venus flytrap. The one big population is an area like maybe twice the size of this room. That, you know, I take people out there without kind of telling them why. And then as they're walking out there, I tell them, okay, look down, because you're just walking on a lawn of Venus flytrap. It's just everywhere. And so during the right time of the year, they die back during the winter. So I have no qualms about harvesting these endangered invasive species and bringing them into classrooms and stuff. But that's an active trap because it actively catches things. There are other types of active traps. Other one you can find around here. Oops, no, I guess I have it later on. The other one you can find around here is these are yellow flowers. I'll show you some pictures of how they do it later on, but they have little spheres on their roots, like thousands of them. They capture microorganisms in the soil in the same way it's an active trap. And so what happens is there's kind of like a basketball with a flap on the side, and if something gets too close to it, triggers little hairs, the flap quickly folds in, and in doing so, it creates negative pressure and sucks stuff inside, and then the flap closes again. And so it's eating microorganisms in the soil. There's some floating ones around here that do the same thing, just in the open water. That's an active trap. They're actually going out and grabbing food. And remember, they're doing this without muscles, without nerves. And in this case, it's almost totally controlled by a really fancy water system. And so there's these hairs that you can't quite see here. We'll see a better picture later on. You have to touch a couple of the hairs, and then it triggers a water pressure system that's kind of built up like a snap trap, that the water suddenly is released and it closes the leaf. And the nerve part of it is that the hairs just trigger that water release, and so it's all vascular. Second way in which insects or carnivorous plants capture insects is with stickiness. And so you may have seen sundews around. They're really common on roadsides throughout much of the world. They're a really common plant worldwide, and they have just these sticky hairs on them. So it's a little hair with a drop of glue on the tip of it that captures lots of insects. And a cooler form of it that we have around here is very, very closely related are these guys, which are called thread dews instead of sundews. And if you get a chance afterward to come up, you can simply take these, and if you put your fingers on them, then slowly pull your fingers away, there are these gooey hairs that come off, like gooey strands. And so you can actually see the glue on each of these things. And the glue's not sugary, and so it doesn't leave your fingers sticky. Your fingers feel perfectly fine afterwards. So it's some sort of water-based sticky glue. But if you look at these leaves, they're covered with little insects. So these things are really good at capturing little insects. It's got to be something pretty small, but there are a lot of little small insects out there. And so it does a pretty amazing job of just capturing really small little insects. So this is a sticky trap. So we have active traps, we have sticky traps, and then we have what I call traps for dumb organisms, and those are pitfalls. And so this is a plant that I work with mostly. It's called the purple pitcher plant, and it just has these big open cup-shaped leaves. And there's a little bit of nectar on the lip on the front of the plant. You can see it kind of looks sticky. It is a little bit sticky. That attracts ants, and ants are just stupid, and so they fall inside the leaf, and once they fall inside the leaf, they can't get out and they drown. And the plant makes digestive fluid, or there's bacteria there that absorbs them. So the same thing would be true with like these guys. These guys mostly attract like flying insects. This is a yellow pitcher plant. And all these occur like within 15 miles of here. And it attracts things that think it's kind of like a flower. It also makes nectar around the top. And once something goes down inside, thinking, well, there might be something more inside, then they just slide down. They're really, really slippery on the inside. And if they start to slide down, since it's mostly attracting winged insects, they can't open their wings to fly out. So that's the end of them. They're stuck there. I think I have some sad photographs of a grasshopper. Yeah, that's a grasshopper stuck down one of these. Great big old grasshopper who's never coming back out again. When you work with these and you're out in the field and you kind of like find some insect who's just fallen in, do you take it out? Do you not take it out? Do you disturb nature by going ahead and saying, oh, that poor spider, I'm going to let it out, or poor grasshopper? Depends on how I'm feeling that day. Some days I'm more on the nature side and think, well, I should let that plant eat. And sometimes I'm feeling sad and thinking, that plant's got lots of food. Let's save this poor soul. This particular plant, when I just showed you, if you look at the top, you can see kind of a ring around it. It's nectar, just around the edge of the leaf. Okay, and so that nectar attracts in dead prey. And so you can just see right around the edge of the leaf right there is kind of glistening some. So that's sugar. It's mostly fructose, I believe, that the leaf makes. But it also has Coniine in it, which is the same poison that's in poison hemlock. And so it's thought that what happens is that the insects will come to this, and the Coniine doesn't really kill them. It just makes them even stupider, and so they're more likely to fall down the leaf and then not come out. So those are our three types of traps. And again, this is the one that makes Coniine. You can see just the little drops on the side. All these make nectar. Nobody has ever looked to see if there's poison in the other nectar. There's a lot that isn't known about stuff. There's a lot of nature still out there for us to explore. We do know that they also exhibit UV patterns. And so if you look at a sunflower and you turn out the lights and shine a UV pattern on them, what you'll find is that this is highly reflectant. And then there are often lines on the leaves like this, telling the pollinator where to go. And so flowers, insects can see in the UV, and flowers and other plants take advantage of that by using it to get insects to do particular things they want. So in this case, this is what the normal looking leaf likes for like this plant right here, Sarracenia lata. And this is what it looks like if we turn out the lights and put on UV. It glows all around that rim to attract insects into it. And so, and again, it doesn't happen to happen during the dark. It's just that insects can see in the UV, so they'll see this in normal light. So there's lots of ways in which these plants are trying to attract in different sorts of prey. Recognize that these are really different carnivorous plants from each other. They're very, very different strategies for capturing things, and that's because they capture very different things. Some capture ants, some capture wasps, some capture bees, some capture butterflies, some capture grasshoppers, like the one I showed you before. All right, so what do I want to trick you into thinking about? So evolution. So these plants occur worldwide, and carnivorous plants of different types occur like everywhere. They're incredibly widespread, except they have highly specific habitat requirements. So they don't occur everywhere in Florida. They occur in bogs in Florida. That's a very limited habitat. But bogs occur throughout Florida, so they occur throughout Florida. And so wherever you have kind of a boggy sort of environment, you can get carnivorous plants. So if you want to study where they evolved or how they evolved, and it's something like, oh, if you wanted to figure out how all these plants that have this sort of flower structure, like a sunflower, where you've got a dense set of flowers in the middle and then flowers making these rays on the outside. If you wanted to study how sunflowers evolved, then you'd take a bunch of different plants and you'd sequence them and you'd make a family tree. And what you discover is that all these things came from the same place on the family tree. And then you can start to ask within that tree, how did this type of a flower evolve? So you can do the same thing for carnivorous plants. You can just kind of take all the different carnivorous plants and a bunch of other plants, sequence them all, and look at the family tree. And so when you do that, you get this and this kind of weird family tree. This has representatives of kind of every plant family you can think of. There are roses in here. There's pines in here. There's kind of anything. These are all the monocots. So all the grasses are here. These are mostly all the dicots. And these yellow areas show you where carnivorous plants are. So when you see that sort of a pattern, that tells you that carnivory evolved a bunch of different times. It didn't just evolve once. So it evolved here, it evolved here, it evolved here, in completely unrelated groups of plants, except that they're all plants. So that's kind of cool. It means it's something that evolved easily. And so there's certain traits that evolve easily. Some of the traits are harder to evolve. C3 and C4 plants is another one that seems complicated, but it's easy. I have a hard time coming up with better examples. Things that are hard to evolve or easy to evolve. But in general, evolution simply requires that there's strong selection and that there's genetic variation. And if you have those two things, then you can get things evolve. Okay, flying. Yeah, flying evolved. How many times did flying evolve? Any idea? No, it's not really active flying, though. That's just kind of floating out there, right? We're talking about wings flying. What other besides birds flies? Insects? What else? And they're not related really to birds very closely, right? Bats, right? And so bats are the classic example of mammals that evolved to fly. So that's something that evolved several times, but probably is hard to evolve. Okay, so we have all these different evolution. These three areas of the 5 all make pitcher plants. So you can even get the same type. And I told you those different types of traps: sticky traps, active traps, pitfall traps. These are some that have pitfall traps that all look really, really similar to each other. So I've showed you kind of this plant that makes those leaves about the right size. They've got a little hood over them. They make sticky stuff. This is a completely unrelated species. This is Nepenthes from Indonesia. And it makes about the same size leaf. It's got a goofy little hood over the top. It's got nectar on the lip, attracts insects in the same kind of way. But it's totally unrelated, except they're both plants, right? But otherwise, they're completely independently evolved the same mechanism to capture things. So carnivory seems to have evolved quite a bit. In fact, there's 3 different types of pitcher plants that look really similar to each other that evolved in different places and from different groups, as is shown here. This is that Nepenthes I showed you from Indonesia. There's one species and one genus of the plant that makes pitchers like this. Same size, same goofy little hood over the top, same sticky band on the front that attracts insects that fall in. But there's only one species and one genus in Australia. And then there's our pitcher plant, the one that I study. Yeah?
I know it's kind of a broad question, but just on average, how much do they need to eat? Like how much nutrients would they need? Like, I don't know, take the yellow pitcher plant, for example.
It's like your house plant at home, right? I mean, if you don't give it as much, it grows slower. If you give it more, it grows faster. If you give it too much, it dies. And so it's hard to say kind of how much does it need. So I can tell you that the ones we've studied a little bit, like this guy, we know it captures about 2 ants a day on average. It feeds on mostly ants. But it only feeds on ants for the first about 5 or 6 weeks that the leaf is open. So the leaf opens up, it captures ants for only about 5 or 6 weeks, and it quits making nectar, and it doesn't attract any more ants. And so it's only the young leaves that tend to capture a lot of prey. And then after that, the leaves are still useful, but they're useful to be photosynthetic, but not to capture more prey. And eventually, then the leaf dies, and the plant makes more leaves. Okay. How did it evolve? It's like, there's discussions you hear people have sometimes about how did eyesight evolve. You ever thought about how eyesight evolves? I'm sorry? Magic. It's just magic, right? And so, what good is half an eye? It's not really good for much, is it? So it's sometimes hard to think about structures and how they evolve because it's hard to imagine kind of the halfway point. So for eyes, a lot of these eyes seem to have evolved as essentially heat-sensitive spots or light-sensitive spots. So like a lot of lizards have spots on their head that's light-sensitive, that's not their eyes. And it tells them when they should bask or where they should bask and get the right temperatures. And so they've been able to show that that's kind of the origin of a lot of things that later on turned into eyes. They became better and better at that. They started then absorbing different wavelengths, and they started evolving into something more like an eye. So in this case, you can imagine that some of these plants simply made leaves that accidentally captured water. And if they accidentally capture water, sometimes things fall in it and drown. And then if you have cells in there that are kind of like root cells, can absorb nutrients, then you might get selection that's strong enough to have a plant evolve to be carnivorous in some way if there's enough time. It requires genetic variation in some sort of morphology. It requires strong selection. And for carnivorous plants, it requires a particular environment that's a really low nutrient environment. We'll talk about in a minute. I do work a little bit with a kind of a maybe early carnivorous plant. And so this is the plant I showed you, the thread dew, captures little dead insects in it. This is a plant that grows all over around here. I typically find them in the parking lot for the old Tallahassee Mall. But it's just a little weed that comes up. It's an annual plant. And on the internodes between the leaves, you get these sticky bands. And the sticky bands catch insects. And so you go, oh, maybe it's a carnivorous plant. We spent a bunch of time growing them in the greenhouse and then sticking little insects on them to see if they make more seeds or if they grow faster. We can't find any effect. So they don't seem to be carnivorous. It appears that they do this to keep insects from climbing up the stem and eating the seeds. And so it's just protection. So lots of plants make sticky stuff on them to kind of protect things from getting into them. And if there's any sort of genetic variation that might cause them to be able to absorb nutrients from this, and enough nutrients it helped the plant, then this could evolve later on into some sort of carnivory. So we're still playing around with this species called sleepy catchfly. But so far it hasn't. We're hoping to show maybe it's kind of a step to being carnivorous, but we've simply shown that it's sticky. Ecosystem ecology. So where do these things live? So they require very specific ecosystems, and I've mentioned bogs. What's unique, do you suppose, about where these things live? Yes?
It's very, very wet, and there's not a lot of nutrients in the soil.
Wet, nutrients. There's one more thing. Probably nobody will guess. Okay, so bogs typically form in an area, a typical bog. It's not really a North Florida bog, but a bog forms in areas that are glaciated, and so you end up kind of depressions in the ground after the last ice age. When was the last ice age? Anybody know? You're getting pretty close. It's probably more like 15,000 years ago. It depends on where you are, right? But and so 10 to 15,000 years ago, you ended up with these depressions. They fill with water, and so it's a great place then for plants to grow because you have some permanent water. But it's not part of a stream system, so the water's just still. It's not moving. You get plants. They grow on the sides. They grow in the water. They fall in eventually. They die over long periods of time. And all those plants falling into the water, growing in the water and dying, give off cations as they rot. As they give off cations, then the water becomes more and more acidic. And so you get really, really acidic conditions. Meanwhile, you have all this plant material that's falling down into the lake in some way that rots. And as it rots, it gives off nitrogen that can be used by other plants. But if you get really high acid conditions, it quits rotting. And so the plants don't rot anymore. And so they just pile up on the bottom of the lake. Eventually, they'll actually fill in the whole lake. But you're left with really wet conditions, really acidic conditions, and their nutrients are there, but they're all tied up in dead plants, so they're not in a usable form. So it's a really low nutrient condition. And we get similar sorts of conditions around here in our big open savanna areas in the national forest that tend to be very wet, really low in nutrients, and somewhat acidic. And so that's what it kind of requires. So we have, this is a typical bog in New England where everything's growing in sphagnum around the edge of the bog. The sphagnum will actually, sphagnum moss is a moss, will actually grow out from the edges of the bog. And sphagnum can grow in these conditions because it's a plant that can fix nitrogen from the air. So kind of, like carnivorous plants, it's got another source to get nitrogen. And eventually that sphagnum will fill in the whole lake. It does make what's called a trembling or quaking bog, which if you ever get a chance to go to one of these, do. It's really cool because you can walk out on the sphagnum. Here, we'll make a little stick figure. Walk out on the sphagnum. If you walk out on that sphagnum, you can actually kind of like bounce up and down. It's like a trampoline. If you bounce too hard, you fall down into the lake, which often has leeches in it, but don't mind the leeches. It's still really cool to do. There'll be trees on it around it that are kind of shaking as you bounce up and down. And it's a great place for carnivorous plants, for sphagnum, for things in the blueberry family can survive on low nutrients, so they tend to be found around these and around our bogs. Orchids survive at really low nutrient levels, so a bunch of plants are kind of specialists for these sorts of conditions, other than just carnivorous plants. Yeah, so here's how things turn into habitats for carnivorous plants. So it's a really kind of a rare habitat, but it's a habitat that occurs all around the world, so we get carnivorous plants all around the world. The other fun fact about this that I like to tell people about is this is where you find bog bodies. And so traditionally, when people have started exploring in bogs in Europe, they find these fully formed human bodies that really haven't rotted very much. They're just kind of, the skin is cured like leather, and you can still look at their stomach contents. Often their clothing is preserved. A whole bunch of artifacts about them are preserved. There are like 1,500 of these in different museums in Europe, so this isn't rare. There are some in Florida, Native American ones from an area south of Gainesville where they've done the same sort of thing. They're of interest to anthropologists because you can study what clothes people wore, what their diets were, things like that. In most cases, these were thought to be criminals. So somebody who they killed because they're a criminal, they can't put them in sanctified ground in a cemetery, so they just throw them out in the bog. Or you want to murder somebody, there's a good place to hide a body, just throw it out in the bog. But it turns out the body will stay there for a long time. They've also found things like big wheels of cheese buried in bogs and other sorts of food because people would use them as like a primitive refrigerator. You know, in the 1400s, you didn't have a refrigerator. But if you bury something in a cold, wet bog that has almost no bacterial growth, it'll stay for a long time. So you can find bizarre things in bogs. Not so much around here. Finally, just a little bit of local ecology then, just to tell you where you could go see these things. Because all these except the Nepenthes, this weird pitcher plant, all the rest of these you can probably find within 15 miles of here. This one occurs further south into Alabama, over into Louisiana and Texas, but all the rest of them you could find near here, even though the Venus flytrap isn't supposed to be here. But the easiest place to see them is to just go out Pensacola Street, which turns into Highway 20, but eventually it takes you over to Panama City. There's a town called Hosford right here, which I should label. It's just a, you won't notice it, there's just a flashing light, but there's a couple of little stores. If you go south from Hosford on Highway 65, it takes you through the middle of the Apalachicola National Forest. And this roadside right now will look like this. These are all flowering pitcher plants. And so, or you'll see these big tall guys, which these beautiful leaves just all along the edges of the woods, just like right in here on the side of the road right there, you'll see them. And if you get out of your car and look, you'll find tons of different sundews and thread dews. You'll find this little pitcher plant, the parrot-top pitcher plant. You'll find all these other pitcher plants there. We have about 5 species of pitcher plants in this area. In the fields where I like to go, we have about 32 different species of carnivorous plants, so the groups that evolve differently from one another. And you can find all those kind of going up and down Highway 65 here. It's pretty easy to do. Here's kind of one of the main habitats are these open grassland areas we call savannahs around here. So you can see these are all pitcher plants down there. And then the other habitat we find them in are what are called ecotones. So this is the edge of a forest, and this is the vegetation around a river. And when you have 2 plant communities come together, that area between them is called an ecotone. It's often different. And so this is the ecotone right here. And if we zip down to that ecotone, my terrible drone photos, then you'll find down in the grass are pitcher plants. And so they occur in these areas as well. Usually at least 3 or 4 species will occur together. The different types are the yellow pitcher plant, this guy here, which is thought to be kind of a flower mimic. And again, it makes poisonous nectar. It captures lots of flying insects. We've done a little bit of work seeing what it eats. We tried playing around with plants by just putting them out in the field. And if they've been growing in the greenhouse, they might have a few insects in them, so we just stuff cotton down it. And then we come back 2 weeks later and we look at everything that's above the cotton we knew is new prey. And we can find things like grasshoppers, a lot of bees, some wasps, a beetle. A lot of the smaller things are ants. So they eat a lot of different things, but they're mostly flying insects that are going to make it up there. There's the parrot top pitcher plant that almost always occurs with the other species. I'm just decimating this poor plant. Should have brought another one. This is called the parrot top pitcher plant because the leaves look like little parrot heads. Okay. And so it's actually, the genus is Sarracenia, the species is Psittacina. The psittacines are the parrot family in birds. And so the hole to get into these is way up in the top right there. Way up the bend, there's a hole. And the hole then, oh, we already had some cut. I didn't have to do this, did I? Yeah. Well, we'll cut new ones too. But if you look in the leaves, you can see that they're downward pointing hairs all the way down the leaf. So once something climbs in, it's going to have a hard time ever getting out because these downward pointing hairs will mean it's a terrible little prison that it will never get out of. Yes?
Can those cuttings grow out?
These? Yeah. No. We propagate these usually not from seed. These things make like rosettes. So I can dig this up and I can break it up into bits and then grow the bits is the main way we propagate them. So it's clonal, but these leaves won't grow. These guys do a little bit better with that. You can take off just branches of these and you can make new plants out of the branches. It's kind of slow. It can be done. So parrot top pitcher plants eat mostly little beetles, but if something climbs inside the leaf because of that like stained glass effect, everything looks like a hole, so they can never find their way out. And again, they eat primarily little beetles. This is the species actually I do research on, which I'll tell you a little bit at the end, but this is the purple pitcher plant. A lot of these are named kind of because of the flower they make. So this is called the yellow pitcher plant, but it's also called the trumpet pitcher plant. This is called the whitetop pitcher plant, but it makes a big red flower. This is the purple pitcher plant just because it makes a big purple flower. So the names are pretty straightforward. I forgot to show this to the other class. So this is the purple pitcher plant. This is the yellow pitcher plant. Okay. And wherever you find those growing out in the forest, you'll also find this. And so yes, it's a hybrid. Somebody's whispering over there. You got it. It's a hybrid. And so you just get, this makes kind of a low purple flower. This makes a really tall yellow flower. Sometimes you have a really stupid bumblebee that goes from one to the other. I mean, normally bumblebees are just going to forage within a type and at a certain height. But sometimes you get a stupid bumblebee, and so you get this thing, which is a halfway plant. It's perfectly viable, so that is, it makes flowers and makes seeds that can grow. And so, but the has characteristics, obviously, of both the leaves. You get this nice kind of in-between leaf. I always tell people, like, this is your dachshund that mated with a Great Dane, and you get that really ugly dog, okay? Except that dogs are considered one species. These are considered two completely separate species, yet we still get these viable hybrids. Usually you wouldn't expect these sorts of hybrids to be viable and survive because it allows you to keep your separate species. This is called a Sarracenia. When you have a hybrid, they put an X in the middle of the name. So it's called X catsbania, and is very common around here as well. What else do we have? Real quickly, other species. There's the whitetop pitcher plant, which again captures a lot of different sorts of insects, but mostly flying insects. Yeah?
How different are the yellow and purple pitcher plants genetically?
Not very.
Okay, so like if they're like super far off, wouldn't that be like almost a feat to see something like a hybrid that would actually work?
So when you make phylogenetic trees for something, so when you sequence the DNA and then you ask, okay, who looks like they're closely related to whom? What you usually end up with pitcher plants is something like this. That is, the branches are really, really short, indicating that these aren't really very different than these. And these from this whole group aren't very different from these from that whole group because you have little short branches. And so genetically, they're not very distinct. And you're right, if they were more genetically distinct, you'd expect the hybrid might be less viable. Still begs the question of why you have these really, really different types that breed true, that you can find everywhere across broad geographic ranges. And then you have this stupid thing that only occurs where you find these two. So you never find it by itself. Seems to be perfectly viable. The flower can vary between the yellow you get from this and the purple you get from this. But it's often brown, kind of a mixture of the two. So why does it still exist? I don't know. There's not strong selection against it, like you might expect for a lot of hybrids. And part of it is because genetically they're very similar. Plant breeders like to breed these things. Somebody just sent me a photograph from Lausanne, Switzerland, from a florist window, where there are a whole bunch of things that look like these that they're selling to mix with bouquets and flowers. I had one of my graduate students got married and had all the centerpieces made with pitcher plant flowers. So you can grow. There are people who grow them, and they grow different colors and types and sizes for the leaves to use them in a floral mixture. So it's an interesting plant. There's the hooded pitcher plant. This guy right here. It occurs mostly to the east of us. It's got a nice flower on it. All these were flowering about 3 weeks ago, 4 weeks ago. This is one of the last ones to flower. It has these nice little hoods, hence the hooded pitcher plant. And it captures mostly what are called wood ants, which is a larger ant, which follows a nectar trail up the side of the leaf into the leaf and then falls inside and dies. But all of them have these sorts of flowers. You can see the old flowers here from this purple pitcher plant, or here's an old flower from the hybrid. They're just done flowering for this year, so I couldn't bring those in. The flower itself is pretty cool too. It has a very interesting structure to it, but it's a basic plant flower with a stigma, a style, and an ovary. In case you had any good botany, you know about anthers and ovaries, and okay, it's got all those bits. It's got all the naughty bits it's supposed to have. There are different sundews. There are thread dews, all different things you could see if you just drive down Highway 65 really, really easily. You can find these bladderworts, these things, the little yellow flowers I mentioned that have little bladders. Here's what the little bladders look like. If you have a good microscope, you can see all these little teeny bladders. And here's a kind of a cross section of one. And so if there's a little insect, that red thing, or a little microorganism like a protozoa or something, if it hits these little hairs, then the doors open inward, and it causes some suction, which draws the animal in, and then the door shuts, and they can't get out, and it dies and rots and is absorbed by the plant. There are what are called butterworts, which most people know just because they have really beautiful flowers, and so they have really pretty flowers that a lot of people like. They're kind of boring leaves. They don't make really fun leaves like this. They're just kind of a slimy leaf that has these curled edges. And insects have a hard time getting off the leaf once they get inside because it's sticky, and then they can't get over the little curled edges if they're a small insect. You have Venus flytrap. So here's our standard Venus flytrap. These aren't animated. But if you look at this cross section, you see all these different hairs, and you also see that the leaf is recurved, which simply means like if you take a frisbee and bend it all the way backwards, it would snap back quickly, right? So these kind of make slightly recurved leaves when they're open. And so when the hairs are triggered, it helps the leaf snap closed more quickly to have those recurved leaves. But mainly it's doing it with water pressure. And so you just have water pressure that builds up in certain areas, creates these little snap mechanisms. And if you trigger two of these hairs, you have to trigger two hairs within like a minute of each other, and the leaf will close. If you just hit one, then it doesn't think it's an insect. So it really wants to know there's an insect there. And once the leaf closes, it'll stay closed only for about 30 minutes if nothing struggles. You have to have some struggling inside of it for a little while, and then it'll stay closed for about a day. Otherwise, it'll open back up in about 30 minutes. And the leaf can only open and close about 3 times, and then it's no good anymore as a trap. It's still good as a leaf. It can be photosynthetic, but it's no good as a trap anymore. And so it's pretty cool. Again, it's this plant that seems to have like nerves and muscles without nerves and muscles. Interesting kind of in-between thing. There's some thought that there's salt channels like nerves that are kind of primitive nerves, but that's about it. All right, so finally, I don't study any of these really at all. I study what lives inside of one species. I have these just because they're fun. They're my pets because I can go give talks. I can use them in teaching. I can go give bar talks. I give them away at bar talks usually. I give away plants. Too bad you guys are too young to go to bars. But there are a lot of different things that live on these plants that are specialists. And so, like this is a moth that only lives on this pitcher plant. And it actually eats the leaves of the pitcher plant, but it also probably pollinates the pitcher plant. So it's kind of a love-hate relationship. There are a lot of spiders that live on these plants. In particular, this spider we've done some research on. The body of that spider gets about like that. Okay, so they can get pretty big by the fall. So with the legs, they're kind of like that. And it's called the green lynx spider. It's very aggressive to capture insects. And they like sitting up on the tops of this yellow pitcher plant. They're very common to find up here because normally they sit up on tops of flowers and they're waiting for a pollinator to come. They specialize on like bumblebees and wasps. And if a bumblebee or wasp comes near, they just jump on it and immediately envenomate it and it dies. And so it's not like they can bite it and then track it down through the woods or something. They've got to kill it right away. And so they have very strong venom to do that. Dr. Okita, who was in this class earlier, is actually helping us study some of the venom on these things since that's what he studies. But it lives up on the top of these and is kind of cool. It changes color when it's on this. It's green, but if we put it on a purple flower, then it's purple. And so the spiders can change color, which we've been playing around with some as well. So there are lots of things that live in or on these pitcher plants. But in particular, I just work on this guy. And I can pretty much kind of ignore the plant. I just treat the leaves like plastic cups because those plastic cups, those leaves, have inside of them this really kind of cool community. So this is a food web, which you've hopefully seen for a whole community. And so usually at the bottom of food web is plants of some sort, something photosynthetic bringing energy in. This doesn't have that. It has instead dead ants. That's where everything's coming in from. And so the dead ants are consumed by a bunch of bacteria. There's like 2,000 species of bacteria. They're consumed by a suite of things that eat bacteria, a little aquatic mite, about 10 common species of protozoa, a rotifer, and then those are eaten by my lion, my top predator of the system, which is mosquito larvae that are filter feeding and capturing all of these things. In fact, to some degree eat bacteria as well. Then there are a couple of other flies that also lay eggs in the leaves, and their larvae interact with this community to some degree too. It's a cool community to work with because it's highly co-evolved. All the things with circles around them only are found in this plant. And they're virtually always found wherever this plant occurs, from here up in the northern Canada, you'll find that whole community. And so all these species are specialists that only live inside of this plant, which is convenient as well. And so this allows me to do experiments. You know, we go back and look at this picture, you can see all the pipettes stuck in the leaves. So when these leaves open, they're sterile inside, and I can put whatever community I want inside. Like I can put in a community that doesn't have the mosquito larvae, doesn't have this. So I should see what are called top-down effects then, because if you get rid of this, you'll get a lot more of these, which will give you a lot less of this, which will give you a lot more nutrients available from the ants. So you'll get these kind of cascading effects we should expect to see. We should dump in more ants. We should get more bacteria, more rotifers, more of everything if we do those. So those are called bottom-up versus top-down effects, and we can study those. We can do things like study corridors. So a big thing in conservation biology is to make better ways for organisms to move like from one national forest to another. As you drive like south of Gainesville, there's an overpass that's just covered with trees and plants. And it's closed off to cars just so animals can go back and forth across the interstate. So we can make corridors. I just send out a bunch of undergraduates with pipettes, and they just suck up water from one leaf and move it to another leaf. And so we can make movement between our communities and see the effects on diversity and stuff. So we can do all sorts of kind of cool little experiments with these. And one of the nice things is that if I wanted to do that with whole communities, like I always give the example of going to Africa, and if I wanted to see if there are top-down effects of lions on Africa, well, I'd have to go out there and shoot a bunch of lions, and nobody's going to let me do that. But shooting a bunch of mosquitoes, everybody's perfectly fine with. They're kind of like saying, yeah, kill more of them. It's good. Get rid of them. Here's what the inside of the leaf looks like. So I'm usually working at about 100x. And so it's a really busy community. So there's a big rotifer running through here, but there's a bunch of small rotifers. There's a nematode. I don't have any mosquito larvae here. They're kind of too big to show up in these. The rest of that is kind of just dead plants or seeds or other things that have fallen into the leaf. And there's a bunch of bacteria in the background here that with a better scope, you can kind of barely make out some of the bigger bacteria in these pictures as well. But that's the community I work on. Sometimes it's been called, little swimming pools of death. Most recently, what we've been doing is kind of using just the protozoa to test ideas about coevolution. And so Darwin had proposed that things evolved to occupy unique niches in some way, and that's how natural selection should operate. And so one of the things he looked at were these finches that occur on the Galapagos Islands, and he suggested that, this one's evolved to eat really small seeds, this one's somewhat bigger seeds, this one really big seeds, and that you won't find like any two that are too alike each other. You'll find like these two can coexist, but maybe these two couldn't coexist. And so he used this to kind of explain the distribution pattern of these birds on different islands. So you'll get some species that can coexist and other species that can't. And it's all the result of evolution. These things evolve to be different. It's hard to test that, right? You can look at these patterns that are consistent with it, but it's hard to test a lot of evolutionary ideas, except in these sorts of communities. In our communities, the protozoa have a generation time of 8 hours. So that means I can get 3 generations a day, 21 generations a week. In a month, I got 100 generations. So I can do selection experiments and actually watch these species evolve through time. And so we've been putting a bunch of them together in different combinations and asking, do they evolve to become more different from each other or more similar to each other in the types of things they eat? So that's what we've been using it for recently. And then last but not least, I also work on a bunch of other stuff, right? A lot of different things. As I was telling the last class, I have a really cool job, right? If you're a research scientist at a university, I teach about a third of the time. I was laughing, talking to Dr. Madden about Mr. Madden about how much he teaches compared to what I teach. I teach almost like, he's teaching 5 courses a day. I teach 1 course a week. And so it's like completely different. About half my time is spent doing research, and about half of it is doing what's called service. But the research part of it is to work on anything I want. Right? I can study anything. And my job is to look at the world around us and figure out how it works. That's what a basic research scientist does, whether you're an astrophysicist or you're a chemist or any of these different fields of science. You're just trying to figure out how the world around us works. Because until you understand how it works, then you can't maybe do other cool things with it. So the standard example is Watson and Crick figured out the structure of DNA. They weren't trying to figure out genetic medicine and how to manipulate genes to cure people. They just wanted to know how DNA worked, right? That's all they wanted to do. And so a lot of what ecologists are doing now that's been really valuable is helping to understand the effects of climate change. By just understanding how a community works, then we can start asking, how is that community going to change if it gets warmer or we get more water, more rainfall, or this island, we've been following vegetation out there for 23 years. And so we can start to follow things like hurricane effects. So we divide it up into different habitats, the foredunes, the things right behind the foredunes, and those stable back dunes. And you can see, for example, in 2004 and 5, when we had Ivan and Dennis, there's a big decline in the vegetation. Or Michael, more recently, there's a big decline in the vegetation. So we can look at effects of hurricanes or droughts or sea level rise. Turns out to be a big factor out there. And so it's a kind of a long-term fun study. We get to go out to the tip of St. George Island all the time and study the plants. I think that's all I got to say. So, sorry I talked too much, but you got any questions for me then?
I want to ask a question.
Are you? Yes.
Do you ever thank you. This is kind of like, do you ever feed the carnivorous plants specifically, or can they always manage for themselves?
All right, so it's a confession I have to make then, okay? In that, we had to grow all these plants in the greenhouse to do these selection experiments I just talked to you about. So I would grow 300 plants in the greenhouse. Plants make a new leaf about every week or so, and when there's a new leaf, so I think there's a new leaf on this plant that hasn't opened yet. So I'm going to open it. I can just take this new leaf right here and I can just pop it open, and now it's open. It's completely sterile inside, so I can put whatever community I want inside of it, right? And that's what we use to do our experiments, is these brand new leaves are sterile, so we can create whatever community we want. So the standard dogma for growing carnivorous plants is you don't need to give them insects. In fact, you'll probably kill them if you give them too many insects. The leaves will just rot and they'll just do badly. So they say, well, all you really need to do is just put a little nitrogen in the soil. You don't need to give them insects. Okay, so that's what I'd been doing for about 4 or 5 years, and my plants got worse and worse and worse. They weren't making leaves fast enough so I could do my experiments. I tried giving them different fertilizer. I tried repotting them. I tried giving them more or less sunlight. Didn't do a darn thing. So finally, I took a bunch of them and started giving them dead insects. They grew great. So why I feel stupid is I seem to prove that carnivorous plants are carnivorous. Yeah. So my experience is they grow much better if I feed them. And I go to the store and buy stuff people use for reptiles. It's just a mixture of crickets and mealworms and little shrimp, and it's freeze-dried, and I just put it in the leaves. I obviously keep some leaves still for my research where we don't put anything inside, but otherwise I fertilize them that way, and it seems to really grow well. Yeah?
So does it like smell bad since the insects
Yes.
are rotting away inside?
Yes, that's the bad part about now feeding them dead insects is, yeah, it smells, yeah. I mean, you're welcome to stick your nose in one of these and give a whiff, but it's pretty bad. On the other hand, they do have natural smells as well that aren't bad. So when we were doing that big experiment with this plant, we had huge batches of them. We're moving to the field. We're putting them in a car, rolling up the windows, driving in the woods, and the car smelled great. So the leaves give off an odor to attract insects too, which is quite nice. And flowers have a little bit of an odor to them. It's nice. But the leaves get stinky, and they also do the same thing that you guys may have studied, which is if you give water too much nutrients, like septic tank leakages and stuff, all those nitrogen going into an ecosystem, what does it do? They study this? About to. Okay. So when you dump a lot of nitrogen into a water system, what happens is the plants go really, really well. But when the plants go really, really well, it then takes up all the carbon dioxide, and you get a bunch of then bacterial growth, and that cuts off all the oxygen. And so you end up with a dead body of water. This is what's happening a lot in the Indian River Lagoon area. And so the same thing can happen in these. If we throw in a bunch of insects, then we eventually end up with a ton of bacteria that are bacteria that can grow without oxygen. They're anaerobic bacteria, and they stink really badly. If you go like swamps out here and muck around in the mud, it smells like rotten eggs type stuff really bad. That's because it's anaerobic bacteria. Too much nutrients, not enough breakdown. Other questions?
So because you kind of mess around with what you put inside of it, have you like noticed a pattern like what bacteria attract like bugs more or something like that?
Yeah, it's a little bit of a problem because there's 2,000 species of bacteria. The bacteria species is a weird thing to think about. Species concept itself is odd. We tend to think of things as being species if they breed true, like in horses versus mules versus or different types of deer, different species of deer, things like that. That doesn't apply to bacteria because they reproduce in such bizarre and weird ways. And so inside an average leaf, there's about 2,000 bacteria. We have done stuff like when we follow evolution of one species of protozoa, we watch to see how the bacteria change, and they do. So we do find certain bacteria associated with certain protozoa. And there's certainly certain bacteria that you get with certain prey types. So if you get something that has a lot of fats in it, then you get like this pink bacteria that makes the water pinkish. But we haven't studied it very much. Yes?
What's kind of like a dream experiment or field trip that you would want to take your students on? And is there like anywhere that has more biodiversity than where you study now?
Well, so I go to a place called Crystal Bog, which is in the middle of the Apalachicola National Forest that I can't tell you about because I'd have to kill you because I don't want people to go there because it's where I do my research. And I think it has 12 species of carnivorous plants in it. And it's just a really beautiful site. It's where I did that one drone shot where it goes all the way down to the plants and the grass. I do take classes there. I was kidding, but most people can't find it once I've taken them there anyway. So that'd be the one thing. I also have to say that right now I have been trying to do a little bit of research in Switzerland. And this is a little bit bizarre, but my species, this purple pitcher plant, I tend to think of as my species, it's invaded Europe. And it didn't really invade Europe. People put it in their gardens because it has beautiful flowers and has an interesting leaf form. It's a pretty plant. And then either it escapes from their gardens or they get extra plants and they just think, hey, let's see if it grows in the bog here. And they just toss them in the bog. And so it's now found a bunch of places in England and Ireland. And in Switzerland, it's found at a whole bunch of different elevations. And so there's plants up at ski resorts, and then there's plants down in the valleys. And so we've been doing some experiments trying to look at kind of climate change effects by taking the plants from the high elevations and moving them to low elevations, so mimicking warming weather of climate change. That's really fun. I mean, it's really beautiful places to work, and it's pretty cool. So that's my favorite place to work right now.
Who else?
Would there ever be like another plant you would want to study, or do you just want to stick with the carnivorous plants?
Well, I study about 72 plants out on the barrier island. If I showed you a bigger view of this greenhouse right now, there's a big experiment going on looking at germination traits in some kind of a sunflower type species around here. There's a big project looking to see whether plants on dunes get their energy from rack, the dead algae up on beaches and stuff. So we think the rack maybe help form proto-dunes. The rack helps capture a little bit of sand, so it makes it more stable. It provides nutrients maybe for plants to start growing that then stabilize the dunes and hold wind and make stuff. So there's a study going on with that. There's been a big duckweed competition study with the class. As a research scientist, right, which most people at Florida State University in science fields are what we call a research scientist. It means about a third of our responsibility is teaching, about half of our responsibility is research, and about whatever's left, a sixth of our responsibility is service type things, whatever that is. But my job, for the research part at least, is to do whatever I want. My thought is I want to look at the world around me and figure out how it works. And that's what a lot of basic research is. Every major invention we've ever had, every major advance we've ever had in science came from basic research. And so, like Watson and Crick, we're not trying to figure out how to manipulate DNA to preserve diseases when they discovered the structure of DNA. They just wanted to know what DNA structure was. And so almost all science starts from just understanding how the world works. I have the coolest job in the world. I'm really paid to just look around me and figure out how the world works. So I can study any sort of goofy plant I want. The caveat is you have to be good at it. I mean, you have to be well-trained. You have to be smart. You have to be able to get grants and publish papers from it. So, you know, when I write a grant for $100,000 at the university, or $1 million, let's say I write $1 million grant, then there's a line at the bottom of the grant. I multiply that $1 million by 53%. And so I actually write the grant for $1,530,000. Okay, that $530,000 I never see. The university takes it, and so the university wants people like me because, or Dr. Okita, or I don't know who else you've had in here, because of those research dollars. So I have to be able to do that. Other than that, I can start working on French fashions. As long as I can get grant dollars, publish papers, I can do it.
Thank you so much, Dr. Miller.
See y'all later.